So, good evening, everyone, and thank you for joining us for our My Macular and Me webinar. Tonight, we are joined by Elena Piotter, a PhD student at the University of Oxford, who will be updating us on her recent project. Her PhD has been exploring treating Stargardt disease using a gene editing technique called CRISPR to correct mutations in ABCA4, which is the cause of Stargardt's disease. As many of you will know, Stargardt is the most common form of macular dystrophy, affecting approximately 1 in 10,000 people. CRISPR is a gene editing tool that works like a pair of highly precise molecular scissors. It can cut DNA at an exact point in the genetic code. In this project, Eleanor and the team focused on tiny spelling mistakes found only in the faulty ABCA4 gene that causes Stargardt disease. By targeting these errors, they aim to correct the genetic code so that the cell can produce enough healthy ABCA4 protein to help macular cells function properly again. Before starting this project, she gained a wide range of research experience, from discovering natural pest killers in soil to working in protein engineering, but she had never worked in eye research. She has an undergraduate degree in international relations with a focus on health and global disease biology, as well as a master's in molecular biology and biotechnology. At the end of the talk, she'll be answering your questions, so please pop anything you'd like to ask in the chat box throughout. We'll answer as many questions as we can at the end. So welcome again, Eleanor, and thanks for joining us this evening. This is such an exciting project, and we're hearing about more and more progress being made in gene therapy. And I know it's something that our audience are really interested in. So I'll now hand over to you to tell us more about the project and if this technique has been a success.
Yeah, thank you so much for having me. And yeah, I'm so excited to be here today. Let me just quickly share my screen and go into presenter mode. Let me know, is that all good?
Yeah, that's all very clear. Thank you.
Perfect. Thank you so much. Yeah, so thank you again for having me. And also I really want to thank you like at the Macular Society and Retina UK for funding this PhD project because without you guys, I couldn't have done this. And so this project focused on gene editing for Stargardt disease. And yeah, thank you all for joining tonight. I really appreciate that you guys are taking the time to be here. So today I'll give a brief background about myself and then the research and the field and end on where the field is at now. So I grew up in between Salt Lake City and San Francisco, California, which are two pretty different places. One is really mountainous and snowy in the winter, where it just actually snowed now, and San Francisco, which is like a much more coastal, chilled out climate. And so I ended up doing my undergrad at the University of California in Davis, which is kind of an agricultural city. So I have a little cow on screen because it's kind of called a cow town just for fun, and did my degrees there. And while I was there, I was doing my first kind of stint in research, doing protein engineering. And so we were looking at like how you can modify aspects of protein to like change how they um, process a reaction. And so that could be like the type of reaction or the rate at which they're doing that reaction. And then how well computer models could predict this. And so that got me pretty interested in molecular biology. So then I went to the Netherlands to do this master's. And while I was there, I did like a first internship looking at biopesticides. And so that's kind of interesting because it's like there's bacteria that live around the roots of plants, and they can produce natural compounds that have antimicrobial activity. And so we were looking at isolating those to see if they could be useful, to prevent against diseases like E. coli and stuff that naturally live on plants. And so then my second project was at Oxford, and so this was with Robert McLaren, and he is quite widely known for bringing gene therapy to the UK, basically for eye disease. And so my project initially was just looking at gene editors, and wasn't super involved in the retinal degeneration aspect of things. But while I was there, I was fortunate enough to be able to watch a clinical trial surgery and really learn about that side of things and about the eye. And I became quite interested in it. And so I was very lucky to be able to continue the research in the lab and to do a PhD. And so now I'll go into some background about the field and some scientific background before continuing. And so, as you know, this research focused on Stargardt disease, where over the course of your life, if you have affected genes, you will start losing central vision, and that can expand outwards into peripheral vision. And so, basically, just to start at the beginning, so to first kind of go over what is a gene. So, as many of you will know, you have chromosomes that are made up of DNA. And so a gene is a stretch of this region that codes for information, and that is through these molecules. So you get GC and AT pairing. So that is guanosine, cytosine, adenine, and thymine. And so G's and C's always pair together, and A's and T's always pair together. And the kind of the process that happens in cells is that you have DNA, which then gets transcribed into RNA, and then you that gets translated into a functional protein. And so DNA kind of looks like a ladder and is often twisted in pictures. If you imagine that being untwisted and cut in half lengthwise, so like where you would be standing on a ladder, that is what RNA looks like, sort of a single strand of that DNA. And on the RNA, you get like three of these bases, so like A, G, T, or C in a row, and that will make one code to make an amino acid for the protein. And so this is kind of the code that functions across all cells to be able to make your necessary proteins to allow for cell function. And so these proteins can kind of vary based on cells, where some proteins are only expressed in certain cells, whereas other proteins are expressed across all cells because they're so necessary for all cell function. And so in instances where something kind of goes wrong in this, there's a lot of like points where errors can occur to then get a non-functional protein. But a big thing with inherited disease is that you'll have mutation in the DNA, which means that subsequently the RNA and then the protein will also potentially not function properly. Well, I guess the RNA doesn't really matter, but the protein will then not function properly, or you get no protein. And then that, of course, then affects cell function. And so one method that has been applied to try and kind of get around this is gene therapy. So here you have a cell, which kind of looks like a balloon in this picture, and a virus on it. This image is actually a bacteriophage, but for the principle of this talk, this is a virus, and you have like squiggly stuff on the inside, and so that will get injected into the cell effectively, and it is carrying a functional copy of a gene that is not working. And once it gets into the cell, it can get expressed. And that then allows for the cell to have the functional protein that it needs. So this has been used for certain degenerations in the eye, including Leber's congenital amaurosis is the famous one with the approval of Luxturna. And I think to date that is still the only approved gene therapy for eye disease. And Robert was working on one for choroideremia, and he's also worked on a few others as well. But those are still ongoing. So the eye is a really great target. It has immune privilege. It is also quite accessible surgically, which is quite nice. And a lot of the endpoints that need to be measured are non-surgical as well, or non-invasive. But also very importantly, because you have two eyes, you also have a control eye that you can use from which you measure any improvement. So you can actually see, or it's more easy to determine whether the therapy is working or not. And here you have a picture of Robert actually doing one of the gene therapy injections. And so for Stargardt disease, there's been a lot of research into making a gene therapy by trying to deliver the ABCA4 gene into the eye. The issue with Stargardt disease and ABCA4, though, is that the gene is too big to fit into the typical viral vector that's used to deliver the gene into the eye. And so, because it has a very small, unfortunately, carrying capacity. And so here there's a picture of a very large cat trying to fit into a very small box, and a lot of it is kind of like spewing out over the side. And it's pretty similar for ABCA4, where if you try and package the gene into this small virus, you often just get a very much shortened version. And so then you get you don't get a complete protein if it is delivered to these cells, the photoreceptor cells. And so that unfortunately is not a viable path forward. So then there's like two different paths forward in how to address this problem, where people are either changing what you're putting into the virus or into the delivery vessel, which is like making a cat smaller, or you can try and make the box bigger. So my research focused on making the gene or figuring out what else we could put into the delivery vessel, whereas a lot of other research has focused on changing the actual package. So there has been research also for Stargardt to use a different virus that has a much bigger packaging capacity, but that unfortunately didn't work. And then there's also a lot of research in making nanoparticles like lipid nanoparticles or other sorts of particles like that to deliver the gene to the photoreceptor cells. And so those and a lot of that research is still ongoing as well. And then there's another version where people have split the ABCA4 gene in half and deliver the front half in one virus and the back half in another. And once both of those coding sequences of the gene have been delivered into the cell, they find each other and they reconnect, and then you get expression of the full length of the gene and the subsequent protein. And so those are kind of the main methods there. And so what I was focusing on is how you can deliver a gene editing tool to actually just correct the mutation that's naturally in your cells. And the way this is done is CRISPR-Cas, which I'm sure a lot, or one of the ways it's done is with CRISPR, which I'm sure a lot of you have already heard of. And so just a quick background, CRISPR was in its modern function was kind of discovered in 2010, but before that, there was a lot of research into understanding what it does. And so it kind of evolved in bacteria as an adaptive immune response because there's specific kinds of like viruses that attack bacteria called bacteriophages. And they will, similar to like when you get a viral infection, they attach to the cells and they inject their genetic material for the cell to then, or in this case, bacteria, to then make more viral particles. And what Cas evolved to do, it was to be able to identify that and cut up that gene so that no viruses could be made, or bacteriophages, sorry. And the interesting part that it did is that it can take us those small fragments and insert them into the bacterial genome so that the next time there's a bacteriophage infection, it can quickly recognize that that is an invasive thing because it has that little sequence as kind of like a memory for that. And so it will very quickly be able to re-identify it. And these are then passed on to the bacteria after that. And so what Jennifer Doudna and Emmanuel Charpentier discovered is that the Cas actually can work in many, many cell types, and that you can just give it a piece of small DNA to find a target and act on that. And that is what they ended up winning the Nobel Prize for in 2020, I think. And this kind of revolutionized like what you could do with in terms of gene, excuse me, gene editing. So there's just a part two of understanding like what the components are. So you have the Cas9, and then there's another variant called the Cas13. So the difference with these is that Cas9 targets DNA and Cas13 targets RNA. And then there's also a PAM site, which is a necessary component only for Cas9, which I'll get into a little bit more detail. And then also one note is that Cas can come from a lot of different bacterial species. So every type of bacteria will have its own type of own Cas enzyme. And so these range in size. And so the most common one used for gene editing is the Streptococcus pyogenes, which is right here. It's also like quite a large one at 4,100 bases of like that DNA. Um, Whereas Staphylococcus aureus is a much smaller one. And so that's the one I focused on because then you would be able to deliver it in an AAV capsid, which is like the viral vector delivery. So the way it works is that you have the Cas enzyme with the guide RNA that you designed for that specific target region in the DNA that you want to target or yeah, address. And so you get this like enzyme fused to the guide RNA and together it will like these two components will go through the genome and kind of track along it. And when they find a PAM site, which is like specific to that Cas9, the DNA will unwind and it will see if the guide RNA can bind. And if it can't, it just keeps going. But if it can, then the guide RNA will bind and the Cas9 in its original format would just cut the DNA. And then it would render that gene, for example, shut off, and it won't work anymore. And so this was really revolutionary and very exciting because for research, it was awesome because you could see do a lot of functional tests on like what different genes did very quickly. But clinically, it was also quite interesting because, like, for example, there was research where they were trying to like shut off all HIV inserts in cells to like try and like effectively cure HIV, for example. Um, and so there's like a lot of therapeutic applications for it as well. But a lot of work that ended up happening was like modifying this Cas enzyme to see if you could do other editing stuff with it. So there's a lot of variations that emerged from this. One of the most exciting being where other smaller proteins would be fused to it that would then do the actual work, whereas the Cas9 would be kind of more the way of bringing that enzyme to the target area using that guide directing mechanism. And so one of them that emerged was base editing, where you can actually just change one base, one specific mutant base, which is quite amazing. And so just to recap, so you have the DNA that goes into the RNA or gets transcribed to RNA, which gets translated into protein. And so I was trying to compare targeting the mutation in both the DNA first and then also the RNA, or just like comparing those two methods and seeing which was more efficient to then get a functional protein. And so yeah, also just to reiterate that the G's are always paired with the C and A's always paired with T. And so some very common mutations that arise are G to A mutations. And so one of the DNA base editors, they can correct those mutations and swap out an A to a G. And so the way it works is very similar to the Cas9, where you have like a deactivated cat or yeah, impaired Cas9 that is fused to this RNA. And then it's also fused to this deaminase protein. And so this all kind of goes along the DNA until it finds the PAM site and the guide or the target area, and it will unwind, and then the deaminase domain can make that base change. And the nice part with this is that because the Cas9 is impaired, it doesn't cut the gene. So that is kind of the important, or it doesn't cut it twice, which is quite important. So you get this nice A to G correction of a G to A mutation. And so similarly with RNA-based editing is that you get this Cas13 fused to a deaminase domain as well, with the guide RNA all complex together. And so they'll also just go along the mRNA until they find the target area where it can bind, and then the selected base can be corrected and to the non-mutant version. And so then in both instances, you would end up with downstream corrected protein. So what I ended up finding is that so initially, like I tested this a lot in just in cells on plates, and that is a very specific environment where you just have a lot of the same cell, and you have quite a large area where you can put the molecules that you're delivering and the proteins and everything. And so in cell models, you get like pretty high editing, and that worked well for both DNA and RNA-based editing, where across a lot of mutations that I tested, and tried to correct, like for RNA-based editing, you could get really high editing rates of like 50 or 60%, which was pretty incredible. And DNA-based editing was a bit lower, but still like very consistent. But then what often happens is when you move into more complex models like mice, is that things like efficiency rapidly drops off. And so with RNA-based editing, it didn't really end up working at all, which was quite interesting. Whereas with DNA-based editing, I did find that there was editing in the mice. And so that was just quite amazing to see. And so, yeah, it's just like a very exciting outcome. And so this isn't to say that RNA-based editing doesn't work at all. It didn't work for me trying to target what I was targeting in the mice. And same with DNA-based editing. Like these systems can be optimized so much. Um, so what this would look like downstream as a gene therapy, or would be also as a gene therapy, where you deliver the CRISPR in a virus, or a different kind of delivery vessel. But for the eye, AAV is kind of the most common method because it works really, really well, and specific to photoreceptors critically. And so this has been, and base editing specifically, has been used in people quite successfully. The most kind of exciting recent one is a baby KJ that had a CPS1 deficiency that can cause, severe issues. I'm not super clued in to the details of the deficiency, but yeah, baby KJ was treated with a base editor and corrected the relevant mutation, and he's now happy and healthy at 2.5 years old, I believe. And so the field of base editing has a lot of promise, and it's very, very exciting to see where it's going. There are a lot of considerations, though. So when designing like which mutation you can target, some of the things that have to be considered are the PAM sequence, because, for example, the DNA base editor won't be able to target an area where there's no nearby PAM site. Fortunately, there's been a lot of evolution of the cases that are used, and also the like how they identify a PAM site. And so this has become quite flexible and has become less of a limitation than even at the start of the PhD 4 years ago. This has become a much different field already, which is kind of crazy to see how quickly it changes. The way, delivery to cells. So yeah, figuring out like which delivery method to use is quite a big one. And you want to try like the amount of cells that are delivered to is a very critical factor. So the more cells, the better. The efficiency. So for DNA base editing, you need a lower efficiency, or it's thought that you need a lower efficiency, because when you correct the DNA, every mRNA transcript after that will have the correction in it. So all of the protein subsequently will also be corrected. Whereas if you're targeting the RNA, you have to continue. It's like all of the DNA will have that is being transcribed to RNA will have the mutation. And so there's a lot of transcripts that have to be corrected for the same effect. So for RNA-based editing, you need a much higher efficiency. And so this is definitely something to consider, particularly in something like Stargardt, because the outer segments of your photoreceptors, they constantly regenerate. I think the renewal is like every 10 days. And so that means there's a lot of ABCA4 being produced because that's where it's found. And so you kind of need a high efficiency for something like that. So then another consideration is off-target effects. So if sometimes the guides, they will bind to areas that are not the target region and make a change. And that is, of course, not wanted, especially because it's not known, what the side effect of that could be. And so that's certainly another consideration for DNA versus RNA-based editing, because if you're targeting the DNA, that change would be permanent, whereas RNA is kind of just, it's made from the DNA. And so if you have an off target there, it might just degrade, or you would just have like a few transcripts that don't, that have this other variant, but it might not be detrimental. That being said, depending on the rate at which that happens, it could also have a detrimental effect. So that's just another consideration when trying to decide what to target. And then yeah, and then the length of efficacy. So for DNA-based editing, ideally you would have the Cas in the cells, they would make the edit, and then we would find a way to shut it off so that you don't have this risk of off-target effects. But there's still a lot of research going into that right now. That's not quite there yet. Whereas for RNA-based editing, you want continued expression because only the transcripts are being corrected. So the mutant version is continually being produced and needs to continually be corrected. So there's just so many things to consider when doing these gene editing yeah, methods. But it is, the field just progresses so rapidly. So a lot of these things just get addressed or yeah, are already just in like a very amazing state of function. And so this is particularly important for inherited disease because about like greater than 50% of the mutations that are single base mutations are G to A and T to C mutations that can be targeted by these DNA and RNA base editing tools, which is very, very exciting. For ABCA4 specifically, 63% are targetable by these base editors of the 1,200 known pathogenic mutations that can lead to Stargardt disease. And so, because it's evolving super quickly, there's also other methods that have been developed to target not just these types of mutations. So you have prime editing, which can correct basically any kind of mutation in terms of single base, and then also small insertions and deletions, which is quite amazing because that covers the vast majority of disease-causing mutations. However, some of these systems are quite site specific, and especially you find this more with prime editing because it's a slightly more complex system. So the more moving parts there are in a system, typically the more considerations there are in terms of like how well it will work. So yeah, that's also something to consider. So some of the ongoing ABCA4 gene therapy clinical trials right now that I think are very exciting are the dual vector approach, which has been optimized from previous ones. And so these are being done by Avant-Garde and Splice Bio. And they work by having, yeah, the two halves of the ABCA4 gene both delivered to the same cell, where then the two halves of the gene can come together and then express the full-length protein. Um, and so the thing with like the dual vector approach for any kind of gene is that there's a lot of things that have like a lot of moving parts that have to come together. So you need a high enough dose that will then make sure there's enough like of each of the viruses that they end up in the same cell where the two halves can then find each other. So, you know, there's like a lot of steps of things going right for that to work. But it will be very, very exciting to see what the clinical trial results will be in some years. And then Acidian Therapeutics is targeting RNA, but it is not using a base editing technology. It will be finding a way to splice in the first or second half of the ABCA4 gene to the naturally occurring transcript, which is really exciting because you get native expression levels and of the ABCA4, and it will be able to address, you know, like both of these or all of these methods enable you to address all. It's not mutation specific, I guess, is what I'm trying to say, which is just a very much more efficient way of yeah target or affect yeah fixing the gene. So yeah, that is the presentation. Thank you so much for listening. And yeah, thank you again to the Macular Society and Retina UK for funding this project and to the Macular Society for having me today. I really, really appreciate that. So
Thank you so much. That was a absolutely fascinating talk, and I know you already scratched the surface at the topic, but it's interesting. It's sort of as a layperson, you hear about gene editing and you think, oh, it's straightforward. It will just go in and fix it, and you've started to highlight some of the many ways. It's a lot more complex than that, but really interesting project. Thank you.
Yeah,
Um,
thank you so
and
much.
and and just a reminder to everyone, if you've got any questions, please do type them in the chat bot and the chat box, and we will get through as many as we can. As a starting question from me, which is probably one you get asked a lot, and it may be impossible to answer, but I'm going to ask anyway. You've talked about sort of the progress that's being made and the pace of that progress, but also lots of challenges and lots of things we've still got
Yeah.
to answer. I mean, how long do we feel this might be before potentially we may see this?
Oh, I have no idea, honestly.
I know
I
it's an unfair question.
Yeah, I feel like it's also one I feel like I shouldn't comment on because you don't want to
Yeah,
like but I do think there's a lot happening in the field. I mean, there's also the small molecule clinical trials that are happening for Stargardt with Belite and Alkeus Therapeutics that are also very exciting. And just a ton happening in the gene therapy space. So I do think, there's there's just a lot of promise there, I think.
a
It's
lot of
more
fun.
just, I think gene therapy is kind of notoriously fickle and very
Yeah.
difficult to get it to work well. So, um,
Yeah, I mean, it certainly comes across as difficult, and as I say, you've only just scratched the
yeah.
surface. We've had another question, which may be a challenging one. This may be an easy question to answer. What would you see gene editing as accomplishing? Is it a cure? Is it about reversing sight loss? Is it about stopping progression? What does
Yeah.
the success look like?
Yeah, that's a great question. Yeah, sorry, I feel like I should have touched on that in the presentation itself. But I think with this specifically, I think the idea, especially with DNA editing, is that you would be able to kind of like prevent the disease if you administered it early enough.
Yeah,
And given that, with Stargardt, it like typically like really starts progressing a lot in like your 20s and 30s onward, there's like quite a long treatment window where you could, administer it early enough. Um, that being said, it's like once photoreceptor cells start degenerating, like even if you edit those, you wouldn't be able to get that function back. So you want to administer it before that happens. And so the, I guess the editing rate is like the main determining factor of like what the treatment effect is, if that makes sense. Like how much you will slow progression and how much like whether it's curative or not, or whether you're just like slowing onset. And so I think that's something that still needs to be like optimized quite a bit. And
but
that
when you say curative, you're talking in the concept of things not getting worse or preventing vision loss. It's not about restoring.
I think that's kind of the ideal scenario. Yeah, is that you would administer it early enough, that you would be able to prevent it from happening. But that is also largely dependent on like how, um, just how widely the virus you or yeah, how widely you can get the gene therapy throughout the retina, right? That's not only dependent on like the efficiency within one cell. Like it has to get to a lot of the cells for it to work, if that makes sense. So yeah, there's a lot of, I think, excitement there. But yeah,
Yeah, wow, fantastic. And is this likely to be one treatment or multiple treatments over a
it would be
number of
one.
years? One
Um,
treatment.
yeah, you wouldn't be able to re-administer, like at least by viral delivery, because it wouldn't be effective after the first delivery, or at least it's thought to be that way. So yeah,
Wow. So
yeah,
that
yeah. So
is
that would be really exciting, that you would just need one injection or one dose, and then it would work. But yeah, I think. I don't know how close that is. Um, but
yeah,
yeah,
and you've mentioned, lots of sort of different techniques all being pursued by different research groups around the world, presumably. Um,
yes.
And your research is focused on Stargardt. Am I right in thinking that, there are similar possibilities and similar options for a range of other different
Oh yeah,
inherited conditions?
there's tons of research in this space for all kinds of, not just eye, just like everything. There's a ton of research happening there, and it's all super, super exciting. Um, but yeah,
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 fantastic.
it's very cool.
I'm just seeing we're getting more questions coming in. I'm not seeing any more.
Yeah,
So
that's all good.
it might be we have given people a really good overview of your research. So thank you so much. I'm just going to pause for another moment or two just to check if anyone's got any questions they want to put to you. Um, I don't know if this is something you'll be able to answer, but Martin has asked, what's the most advanced clinical trial, I guess, using these sorts of techniques? I mean, by the sounds of it, the work you're talking about is in mice, but these
Yeah.
sorts of techniques, how advanced have things become?
Um, well, so I guess like for base editing generally, there was the one case of baby KJ, um, where it has been successfully used in a person. Um, I don't know outside of Stargardt, about base editing specifically. Within Stargardt, the clinical trials I mentioned are all in phase one, for gene therapy. And then the small molecule ones, the Belite Bio one, I think just passed its primary clinical endpoints for phase three, which would be really exciting. Um, and so that is like a small molecule that basically um, inhibits vitamin A absorption, so it can slow down the progression of Stargardt, which is very, very exciting. And so I think they're I'm not 100% sure on this, but I think there is something about applying to the FDA for approval in 2026, as far as I'm aware, which would be incredible because I think would be the first treatment for Stargardt.
Yeah,
Um, but I think they're still releasing like other clinical endpoints and stuff like that. So I'm not like 100% on that.
yeah, but that's a small molecule therapy you're saying rather
Exactly.
than
That's not gene
proper gene
therapy.
editing. Yeah,
Yeah,
so
the gene therapy clinical trials are all the ones that I mentioned, like the Avant-Garde Splice Bio and Acidian are all phase one at the moment.
yeah. So that's the earliest stage of work and a long way to go before it's in routine use.
Yeah, yeah.
And my colleague has posted a link to find out about more about this research and other clinical trials as well. We've had a question from John. Can these tools be used for treating macular degeneration at some point?
Yeah, I think in terms of like age-related macular
Yeah, I
degeneration.
assume that's what they're referring to. Yeah.
Yeah, I think it depends on the cause of AMD because my understanding is that AMD can be caused by a lot of different factors. It's not only inherited. And so these tools are useful for inherited degenerations. So if the cause is known to be, macular degeneration due to a faulty gene, then yeah, you could. But if it's like unclear whether what the cause is, then I think you wouldn't just be able to, make a gene therapy if you don't know the the target.
Yeah, fantastic. Well, thank you again so much. And I found that
Yeah.
absolutely fascinating and
Thank you so
really
much.
interesting to hear, what's really at the cutting edge because these kind of techniques are still so new, aren't they? And just as I was wrapping up, we had another question, so apologies for jumping the gun there. Is there a way to fix existing damage by Stargardts to restore vision?
Ooh, I don't know. That I think might be an ophthalmologist question.
Yeah, and I think the answer is not yet.
Yeah,
But yeah, we should have said at the start. So obviously, Eleanor's not a clinician, so that probably is one for a clinician. But unfortunately, not yet. That's clearly something we'd like to see. But it's
yeah,
it's always easier to prevent sort of further loss of function than it is to restore things. Unfortunately,
yeah. I think the research for that is centered a lot on like stem cell therapies and stuff like that to put like a new layer of cells. But I'm not super familiar with all like how that works exactly and where that aspect of the field is at.
yeah, we'll look to see if we can have another speaker later in the year who can talk about kind of broader options around Stargardt's treatment, because I know there's lots of interest in a whole range of different therapies. Well, thank you again, and
Yeah, thank you.
been lovely to hear from you. And good luck with whatever comes next in your research. And have a lovely evening, everyone. And I'm just forgive me, I need to give you the time and date for our next talk as well. So our next talk will be same time, so 7 p.m. on Tuesday, the 17th of March. And we'll be joined by Dr. Katie Curran, who is a research fellow at Queen's University in Belfast, who'll be discussing a new clinical trial to explore whether L-DOPA, a medicine already used safely for Parkinson's disease, could help people with wet AMD. So a very different topic, but hopefully a really interesting one. So I hope to see lots of you there next month. So take care, everyone. Have a lovely evening, and all the best. Bye.
Thank you so much for having me. Bye.